Strange pits on ancient teeth could reveal clues about human ancestry. Tiny pits in 2 million-year-old tooth enamel may offer insights into the evolutionary relationships among our early human relatives. For decades, scientists have puzzled over unusual clusters of pits found in the dental enamel of Paranthropus, an extinct genus of early hominins. Now, new research suggests that these pits are likely genetic in origin rather than the result of disease, potentially making them a valuable marker for tracing evolutionary lineages within the human family tree. Teeth preserve an incredible amount of biological and evolutionary information, said Ian Towle, co-author of the study and a researcher at the Paleodiet Research Lab at Monash University in Australia. This specific type of pitting might turn out to be a unique marker for certain evolutionary lineages, helping us identify fossils. Published in the July issue of the Journal of Human Evolution, the study describes uniform, circular, and shallow UCS pits in the thick enamel of Paranthropus molars, an unusual and distinctive pattern. Enamel development can be disrupted by environmental stressors like malnutrition, typically causing enamel defects in the form of lines or random pits. However, UCS pitting appears more structured and consistent, hinting at a genetic origin rather than environmental damage. To determine which early hominins had UCS pitting, the researchers examined dozens of fossilized teeth from individuals who lived in eastern and southern Africa between 3.4 million and 1.1 million years ago. They found that UCS pitting was common at South African sites associated with Paranthropus, with roughly half the specimens showing the distinctive marks. In contrast, UCS pitting was rare or absent in other ancient hominin species. Among more than 500 teeth from Australopithecus africanus, another early hominin from South Africa, there was no compelling evidence of UCS pitting. This suggests that South African Paranthropus did not descend from A. africanus. However, some East African Australopithecines did show signs of UCS pitting, hinting that Paranthropus may have evolved from them instead. A few UCS-like pits were also observed in teeth from rare species in the Homo genus, such as Homo luzonensis and Homo floresiensis, small-bodied hominins that lived in Eastern Asia around 200,000 years ago. The presence of UCS pitting in these species could suggest closer evolutionary ties to Australopithecines than to other Homo species, Towle noted in an article for The Conversation. However, Towle cautioned that the limited number of examples in the Homo genus makes it difficult to draw firm conclusions. Further research is essential before UCS pitting can be confidently used as a taxonomic marker in hominin studies, he said. One promising direction is paleoproteomics, the study of ancient proteins preserved in tooth enamel. This technique could provide deeper insights into UCS pitting and help determine whether it was more prevalent in male or female individuals. The study suggests that UCS pitting may have been a common genetic feature throughout the evolutionary history of Paranthropus. Our research shows that even tiny surface details, like pits or dimples, can be incredibly useful for understanding the biology and ancestry of early human relatives.